So now we will start with epigenomics and cancer. So we know a little bit about epigenetics now, right? DNA methylation, histone acetylation, deacetylation. So this is just a cartoon showing here that many tumors are cold tumors. So what happens is that cold tumors are those tumors which don't respond to any therapy. So they become very resistant. Sometimes what happens is that it is easier to target more aggressive tumors than the non-aggressive ones, because non-aggressive ones are slow growing, but they are very resistant to therapy. So if you can take a cold tumor and you can make them a hot tumor, then more targets will be exposed and you have more chance of treating that one. So that's what this means here. And sometimes epigenetic drugs, that means the drugs which modify the epigenetics, can be useful. So epigenomics means you are looking at, whenever there is this omics word, that means you are looking at it globally in the whole organism or in the whole cell. So this is a cartoon here which shows that this man is a happy mind. This child is a happy child initially, and then early life crisis, some kind of abuse, whatever abuse it is, that created huge adverse condition. And those adverse conditions led to CpG methylation, so epigenetic changes in that body. Okay, so the DNA sequence is still the same. You can imagine the twins were born. This one child was raised in very happy environment, right? And he became a very happy going person, very successful. This child, same intelligence, identical twins, but he faced a lot of adversity, childhood trauma, things like that. So his brain chemistry changed in terms of epigenetics. DNA is exactly the same, but epigenetic modifications are different. So when this person so you know, we all face stressful conditions, okay? I know of a person, the more stressful condition this person faces, the more happy she becomes. So her boss is very abusive and she tells me a story. And when she's telling me the story, she's laughing. And then she's laughing more. And then she's laughing more. So she said, you know, Didi, this happened. अरे भैया तो मुझको पहले बता देते, मैं तैयारी कर लेती, क्यों मुझको 1 मिनट पहले बताया? So every stressful condition is a joke. So she knows how to deal with stress. She's never stressed. And I always tell myself, if I face the same situation, I'll be super stressed. Imagine you're asked to teach a class 5 minutes before the class starts, and you have no idea what to teach, and they throw you in the meeting without telling, oh, speak in front of 100 people on this topic, and you don't know anything about that topic, and that person knew about that topic 10 days ago, but he's deliberately not telling you to put you in that stressful situation. We all will be stressed. This woman not stressed. She said, all he needed to give me 20 minutes, I would have prepared, right? And she starts laughing and laughing, and now what happened? After a year of this kind of abuse, now this person is actually a friend, because he knows nothing will perturb that woman, and she doesn't have any enmity towards this person also, because she's genuinely a happy person. But this person who has faced a lot of trauma, he has so many stress receptors in his brain that small stress is amplified in his brain. And you know, eventually that person cannot handle that. So this is due to epigenetic changes. It could be due to epigenetic changes in the brain. So that is why we all react differently to stress situations. Okay, so epigenomics is tightly regulated with cancer. Many people believe that, you know, if you make epigenetic drugs, you can really change cancer, okay? And epigenomics, okay, that promotes cancer in many different ways. Epigenomics changes can lead to uncontrolled proliferation. It can lead to uncontrolled metastasis. It can prevent cell death. It can change immunity. This is true. When we are stressed, we are more susceptible to diseases that you may all have seen. When I'm stressed, I get cold, I get this, I get that, right? Oftentimes we have fever because we cannot deal with stress. So our immune system goes down. So it evades immunity. It induces angiogenesis. So it promotes cancer in many different ways. Okay, so this is This is the part of epigenomics. This contributes to cancer. Okay, so epigenomics, so you all know about different kind of modification. So I barely told you about DNA methylation and histone modification, but there are other epigenetic mechanisms also that contribute hugely to diseases. And these are nucleosome positioning and non-coding RNAs. Okay, these days these non-coding RNAs, they are becoming really major players in human physiology and pathology. Okay. Okay, so so epigenomics is also called as the software of the genome. So what is the hardware? The DNA sequence. That's the hardware, and the software, which is the epigenomics, is the one which controls how DNA expresses RNA. Hey, no cell phone. Okay, in my class. Okay, so the epigenomics control which genes will be expressed. So DNA sequence is identical, but that is controlling which proteins are going to be made. Okay, so just to remind you once again, so this is here that many different factors. Okay, many different factors control epigenomics. So you may have heard that when women are pregnant, they said in some culture it is very important, especially in Native American culture. So you guys know who are Native Americans, right? You all know Native American culture. So Native American people are spiritually very advanced people. Okay, so in their culture it is essential that the pregnant women do not face any stress. They go out of the way to keep pregnant women happy and stress free, because if the mother is stressed, it affects the child in the at that stage. So that is what it is showing. During pregnancy itself, those stress things can be absorbed by the child, and that can lead to lifelong problems in future. Okay, new childhood trauma is a major. I was reading it today in news that Bob Reiner was killed, right? The famous director of Hollywood was killed by his own child. He and his wife were killed. So he actually made a movie on this thing, and he once in an interview famously said, he was a very nice man. So it's very unfortunate that he was killed by his child, and the child has drug problem. So he did his best to help him out. But he very famously once said that all the bad people, like who are bad people, who kill other people, or who are violent people, right? They have all suffered some kind of trauma in their childhood. If somebody has a very happy childhood, they are not likely to develop into a violent person. So it is very important that, the children have normal childhood, and, we cannot control it because, it is your life circumstances. Many times parents cannot control the circumstances also, despite their best efforts. But childhood trauma can have long-lasting effects. We already talked about all these drugs and aging, physical activity, exercise really helps you, right? And once cancer starts, then, it is due to genomic problems. It could be epigenomic, it could be environmental thing which affects genetics and epigenetics. Okay, so this you already know. See here you have more. Look at this thing here. So in this small area, you have 1400 nanometer mitotic chromosome. Okay, now we open it up a little bit. So now this is the ordered structure. Now we open this structure, it looks like this. We open it further. This is called the 30 nanometer fiber. Okay, we open it more. Now it is beads on a string kind of structure. Now when remove all the histone octamers, now this is DNA, and now you see it there. So this is extremely ordered structure. Okay, so let's focus on this part here. So this is how it looks like. Some part is very tightly closed, some part is more open. So this is more likely to make RNA than this part here. When it is methylated by histone methyl transferases, so remember this is the protein part. So when it is methylated, it is closed, it will not make RNA. Once it is acetylated, now it will make RNA. Okay, so DNA methylation, histone modification, chromatin remodeling, and non-coding RNAs, they control. These are the major mechanisms of epigenetics. Okay, so I already told you about histone modification and methylation. This opens up, this closes it. Okay, so and this is the modification. So this is cytosine is modified, and once it is, this is histone, this is DNA, this is histone, protein modification, both methylation. DNA methylation also closes, histone methylation also closes. Okay, this is cytosine, and then this is the promoter region. So once it is methylated, it is closed. Now RNA polymerase cannot bind, and there is no transcription. Okay, so now you can imagine there, if there is a tumor suppressor, then this is going to get methylated. So tumor suppressor, hypermethylation. Now it is not going to be expressed. If this is not methylated, it is going to be highly expressed. So oncogenes, it will be hypomethylated. Okay, so look at these things here. By looking at this pattern, you can tell which one is a tumor suppressor, which is a tumor promoter. So APC is hypermethylated. So according to you, it is a tumor suppressor or a tumor promoter? Raise your hand if you think it is a tumor suppressor. It is a tumor suppressor, right? Because it is hypermethylated. KRAS, oncogene, right? So you can see cancer cells know exactly which one to hypermethylate and which one to hypomethylate. c-MYC is such a big oncogene, right? Hypomethylated. And also microRNAs. How many of you know about microRNAs? Some of you know about microRNAs, right? So microRNAs are the one which control which RNA will be expressing the protein or not. So what they do, microRNAs can bind the RNA directly, or they can bind some part of the DNA and influence the transcription or sometimes even translation. Okay, so microRNAs do not make any protein, so they are called non-coding RNA. Any RNA which makes protein is a coding RNA, because protein expression of protein is also called coding. So any RNA which does not make protein is called a non-coding RNA. And microRNA influences gene expression in many different ways. So that is why they are not changing the DNA sequence, but it is influencing that. So it is considered an epigenetic mechanism. Okay, so this is DNA methylation. If it is unmethylated, gene is on. If it is methylated, it is off. And because of that, they can control oncogene expression, they can suppress tumor suppressors, and they can lead to plasticity and therapy resistance. So epigenetic changes can occur which can make a cancer resistant to therapy. I will ask you now, give me one mechanism by which epigenetics can lead to therapy resistant. Can anyone tell me? Again ask the question. So tell me one mechanism, one epigenetic mechanism by which a tumor becomes resistant to therapy. So it was responding to therapy before, and then some epigenetic mechanism occurred and it became resistant to therapy. So tell me, make up any one mechanism. All of you have enough background, just give me any one mechanism. The important thing is that no matter what mechanism you make up, right, it exists in nature. because nature is so diverse that you can be 100% sure that that mechanism exists somewhere, even if you think you are making it up. So I will guide you. So remember methylation, DNA methylation, off switch, histone methylation, off switch, histone acetylation, on switch, right? So keep these three things in mind to make your life simple, and then tell me that there is a tumor which is responsive to stress, responsive to therapy, and then it stops responding to that therapy. So I can say one non-epigenetic thing, oh, that protein mutated, it no longer, the drug that you were giving no longer binds there, and so it is resistant. So I just gave you one mechanism which is due to mutation, but it is not epigenetic mechanism, right? Because epigenetic has nothing to do with changing the DNA sequence. So I ask you, give me one epigenetic mechanism. Any one of you. So I want you to think in backward. You know retrosynthesis in organic chemistry? So what is retrosynthesis? You start with the molecule and then you break it apart and then you build the molecule based on that. So I want to do a retrosynthesis like of this thing. So suppose you have a tumor is responding to something. So what is that something? Let's go retro now. Maybe it is due to a protein, right? Yeah, ligand maybe or something, right? Now think backward. So it has become resistant to that therapy.
It might have modified its receptors.
Which way? We cannot use DNA mutation because it is an epigenetic mechanism.
Methylation of the segment which was synthesizing that receptor.
Yes, so now this receptor is no longer even there, and you can keep on adding drug, and it has expressed some other protein, it has rewired itself. That is the most important mechanism. Or you can do one thing also, that it has in normal cells, this efflux protein, their promoter is methylated, so they are not expressed. Now they have removed the methylation from those, efflux pumps are expressed, and the drug is pumped out, right? So that would be an epigenetic mechanism also. So you can think of many mechanisms by which it can lead to therapy resistance, right? And plasticity is, again, I will give your example. Like they don't even make the protein anymore. The receptor is absent, and now they are waiting on something else. They make some other protein, and that becomes the driver for cancer. That is plasticity, right? So you already know how to do that. So here this is normal transcription, TSG is expressed. TSG is tumor suppressor protein. And here you have methylated, tumor suppressor protein is no longer expressed, right? Here in normal cells, oncogene is not expressed because this is hypermethylated. They remove the methylation in cancer cells, now oncogene is expressed. So this is what it is showing here. So promoter methylation is one of the major ways they control gene expression. So here it is methylated, tumor suppressor is not expressed, okay? Now let's talk about other mechanisms. Acetylation opens it up, more transcription. This I have already told you. So if you have to block this thing, somebody was asking me how do you block DNMTs, the methyltransferases. So here if you block this methyltransferase, then this is not going to be methylated, and if it is not methylated, tumor suppressor will express, right? Here you have to do the reverse there, okay? So these two DNA methylation, and what is chromatin remodeling? So I think I have included one figure here, let me see. So look at this structure here. So this is heavily modified. Now once, this is really tight structure. Now once you add the acetyl group here, so sorry, this round thing, this is methylation, and this pentagonal thing, this is acetylation. So when it is acetylated, it is open. When it is methylated, it is completely closed. This is what it is showing. Now look at the structure here, okay? So histone is modified. So I kept epigenetics very simple, but actually there are many more modifications. There are at least 20 different kinds of modification which happens on histone. they can get phosphorylated, they can get methylated, they can get acetylated, they can get sumoylated. Sumo group is a different kind of this thing. They can get ubiquitinated. You know, ubiquitination is because of protein degradation, but there are different kinds of ubiquitinal groups, and some of them turn on something, and they are also signaling, which has nothing to do with degradation. So look at all different kinds of modification: acetylation, methylation, phosphorylation, ubiquitous isomerization, ADP ribosylation, deamination, S-methyl cytosine, and SOMO is not even written here. So there are many kinds of modification, and that is called as histone code. So when there are many modification, it's not as simple as on-off switch, but it is a whole pattern of things which determine like how gene expression is controlled. Now look at the microRNA. So this microRNA, so suppose this is the promoter region from your view, this is the promoter region, this is the gene. So a microRNA may bind to the promoter region and inhibit transcription, right? Or the microRNA can bind to the RNA which is formed and it can degrade it. Or it can sometimes activate also. So microRNAs are also controlling gene expression. Okay. So this you already know, acetylation can lead to oncogene activation. So what about HDACs? So HDACs are histone deacetylases. Somebody asked me yesterday, I don't know who asked me, you asked me, right? So how do you decide? So I am telling you a very simple story that acetylation increases transcription, methylation decreases transcription, right? But things are not that black and white in reality. Sometimes acetylation can also occur in tumor suppressors, right? So tumor suppressors are also expressed more. So it is not that histone acetylation specifically only happens for oncogenes. It can also occur on tumor suppressors. Okay, so in that case, the reverse of histone acetylation is histone deacetylases. So histone acetylase, they add acetyl group, this enzyme removes acetyl group. So suppose histone acetylases, these are called HATs. HATs are adding acetyl group on those proteins like tumor suppressors, and they are expressed. So that's very good. So if you add HDAC inhibitor, then if they remove acetyl group, tumor suppressors are not going to be expressed. So what I'm trying to say is that it is not black and white all the time, that acetylation only occurs for oncogenes. Sometimes it can also occur for tumor suppressors, which is good, right? But it is also occurring to some oncogenes. So how do you decide? The question you asked me yesterday, how do you decide if acetylation is doing both oncogene and tumor suppressor? If you inhibit that, is it good enough? In that case, you have to give the drug and see which one is more effective, like killing the oncogene and killing the tumor suppressor at the same time, because you have no choice, because it is targeting both. So in that case, you have to actually determine experimentally, and it may have some side effect, but in reality it is not crystal clear in every case. There's always some kind of good thing also going on. So that is why it is asking, is it a friend or a foe? Okay, so HDAC can also lead to cell death. It can promote angiogenesis. So there are many things which are controlled by histone deacetylases. We are not going to discuss that. So it is always, you have to weigh good versus evil when you are deciding a therapy. That's the point here. Okay. So now the thing which I did not tell you about was chromatin remodeling complexes. So what happens is that when I showed you this structure here, so in reality DNA is like this. It is like this, right? It is very, very, very complex, and it is extremely folded. Okay, so when you unravel the DNA, right, sometimes if you put something there very far away from the transcription site, that perturbation can have effect here also. So that is what is shown here in chromatin remodeling complex. in this one here. So see, there are many proteins which bind the histones. So look at some of these conditions here. So here it is trying to see, these are histone octamers, right? So there are these proteins which will take out the whole histone out here. So this, they have removed the whole this thing. So obviously it is wrapped around this, and if you remove the whole protein, now there is nothing to be wrapped around, the whole structure will fall, right? So these proteins are called chromatin remodeling complexes. They will come and they will just take the histone away from that, and that it will open. I can see you guys are very tired. We all need a cup of tea to keep ourselves awake. But let's
3 o'clock.
okay, 3 o'clock. We have to survive till 3, then we get tea. Okay? So, okay. So there are several proteins which will bind chromatin structure, which will distort it in this way, that, open it up, right? All kinds of drama is happening which can change expression of gene, which can change gene expression. Okay? So let's briefly talk about these non-coding RNAs. So Pol2 is RNA polymerase, right? The reason it is called Pol2, because in humans, do you know that there are 3 kinds of RNA polymerases, 1, 2, and 3? And RNA polymerase 2 is the one which makes messenger RNA, and messenger RNAs are those RNAs which make proteins. Okay, so they are called the coding RNA. Okay, so once messenger RNA is made, okay, I don't know why it is showing long non-coding RNA. I'm 100% sure RNA polymerase 2 is the one which makes messenger RNA. Do you guys remember which RNA polymerase makes messenger RNA? Second one, right? So non-coding RNAs are, I think, not made by, or maybe they are also made by RNA polymerase 2, because, yeah, along with mRNAs. But this ribosomal RNAs, they are made by partly by 1 and 3. 5S is made by 1, and then other 3 are made by other. So yeah, so this RNA polymerase 2 also makes some non-long coding RNAs. So these are the RNAs which, see, look at this thing. This is mRNA, right? So human mRNAs, eukaryotic mRNAs have a cap at the 5' end, and they have a long poly A tail. You remember? So this is a human messenger RNA. So this is binding this one, and because of this binding, it can stabilize it, depending on where it is binding, or it can degrade it, or it can change the expression of this gene. So this guy has nothing to do with this mRNA. But once the mRNA is formed, it can control what happens to this mRNA, right? So this is the long coding RNA, and this can also degrade this one here, okay? And then this RNA here, this is bound to this one, and then this is bound to another protein. Because this RNA is bound, now a protein binds and it is activating transcription, okay? So this is binding to histone directly, and so you make an RNA which then opens up the chromatin, and then it promotes transcription. Here it is the other way around. It binds, now it calls a repressor, closes the chromatin. So basically non-coding RNA, we are not changing the DNA, but because of this long non-coding RNA, we are controlling transcription, right? So now let's compare genetic mutation with epigenetic changes. Okay, so in this one, genetic mutation is a permanent change in the DNA because you have mutated the DNA, correct? And this can caused by errors in DNA replication or exposure to radiation chemicals, or one can inherit from the parents. Okay, and this is permanent, it can be inherited, and because you have changed the genetic code, it will change the protein formation. So for BRCA gene, they are mutated in breast cancer, they don't work. Do you guys have you guys heard of sickle cell anemia? So sickle cell anemia has mutation in hemoglobin gene, and because of that they are sickle shaped, right? So normal red blood cells, do you know how long of a half-lifetime they have? 120 days, very good. And what is the half-life of sickle cell? 10 days, roughly 10 days, right? You guys tell me, how many days? Anyone remembers? I also remember with confidence 120 days for healthy ones. How many for sickle cell? As I recall, it is 10 to 20, huh?
I think 20 to 30.
20 to 30 days, yeah. So just one mutation and you change it, right? But if you compare it to epigenetic, like DNA modification, non-coding RNA, this is reversible. Okay, this is reversible thing. This is not inherited, right? Sometimes it can be inherited, but most of the time it is not inheritable thing. Okay, and example is hypermethylation, hypomethylation kind of thing. So this is comparing both of the things. DNA is changed, DNA is not changed, right? This is permanent, this is mostly reversible. Okay, this is, it can be inherited or DNA repair mechanism. This is, look at the epigenetics, environmental, diet, stress, drugs. Okay, so I work with a kinase called CDK5, and CDK5 kinase is very good kinase. So when you guys are studying for exam, CDK5 activity increases quite a bit, and that is why your memory increases very high, right? So it's a very good kinase, but if it is always in the high state, active state, the brain cannot handle that much activity and the brain will degenerate and the person will get Alzheimer's. So CDK5 hyperactivation can lead to Alzheimer's. So this activity is ideal. If you have less CDK5 activity, then what will happen? You will have disease, huh? No, neurodevelopmental of this thing. So one person can have schizophrenia. They can have, all these kind of weird diseases, ADHD kind of things. So these kind of diseases can develop if the activity is less. This is normal. And this much activity for a long time, when we have exams, it is slightly elevated. Exam stress is gone, it comes back. So that we can handle, right? But if it goes high all the time, that's when the brain will degenerate. Okay, so but CDK5 people who take drugs, okay, CDK5 kinase is the one which makes the pathways which causes addiction. So CDK5 is responsible for a person get addicted to drugs. So drugs can change the whole neuronal pathways in the brain, and then they will crave for more and more and more drugs. Okay, so so many times that leads to epigenetic changes where the person is really craving for that drug. Okay, so this, in mutation you will get a different protein because you have mutated the gene, but here it controls which gene is going to be expressed and which gene is not going to be expressed. It is not changing the protein itself, but it is controlling the expression level of that protein. Okay, so just see BRCA1 mutation in cancer, BRCA1 promoter methylation in cancer. This is a very good example. You methylate it, BRCA1 is not expressed. BRCA1 is there, healthy, no problem, but you hypermethylate it, it's not going to be expressed. Okay, so let's look at the consequences of epigenetic changes in cancer. We already know silencing of tumor suppressors. We have seen activation of oncogenes. Okay, so go ahead.
How to identify, how will identify that if someone gets breast cancer, so it will means if the gene get mutated, we will find that by any technique. But for means what technique we will use for that epigenetic change methylation?
That is very good question. Okay, so I will even show that thing there. That is very good question. So how will you know it is epigenetically modified? So we have actual techniques which will tell you whether the promoter region is methylated or not methylated. So what you can do, I will tell you very simply, right? So I will tell you about those techniques, I think in this slide, in this presentation only. But in addition to that, what you do, if you do genetic testing and you say BRCA1 gene is there and you can say it is not amplified and you can see that it is not mutated, it will give you all the information. But then you do proteomics and you see in normal things it is expressed and it is not expressed at all, or it is expressed at very low level. But you see the gene is intact. So now you can guess that something is blocking its expression. So you can guess it is probably methylation, right? And then you can figure it out by actually looking at the methylation pattern. And there are ways to do that. And you can look at all the methylation across the genome also. Okay, so these techniques exist. Okay, so you already know this one, so I'll skip. You already know the activation of oncogenes, so I'll skip. Now, I did not tell you about genomic instability. So what is genomic instability? So what happens is that genomic instability is that in general terms, the cancer cells are they're always changing themselves, mutating, in order to become survival of the fittest. But when they do that, obviously some mutations are lethal and they get eliminated. And they don't care about elimination because even if 1 or 2% cells survive, they have acquired those mutations which really enable more and more proliferation and more resistance to adverse conditions, right? So in terms of epigenetics, so why you can gain genomic instability? So when the DNA is replicated, it is randomly modified epigenetically, right? So it can lead to double strand breaks, chromosomal rearrangement, cancer. So a lot of things can happen just during normal process because of the change in histone modification. So the whole genome will be unstabilized, okay? So some of them will die because of that. But in other cases, they will acquire resistance and they will become even more aggressive. So epigenetic changes can change the whole genomic structures, okay? So because of that change, so for example, this is a tumor cell here, okay? And this is, remember yesterday I said that normal cells are pretty differentiated. So if you look at mesenchymal cell, they look like this, right? The more cancerous they become more round. They don't look like the original parental cell. They look like more round, more aggressive kind of thing, okay? So this is a differentiated tumor cell, and then now we gave different kind of radiation. Most of the cells died, and now this stem cell emerged. Remember, 99.99% cells die, but 1 or 2 cells survive, and they are resistant to all therapy, all radiation. These are the stem cells. And because they are stem cells, they now will form new kind of they can modify themselves into different kinds of cells. In some cases, they will have these cells are different from these kind of things. So these stem cells, they have infinite renewal property. So they can keep on making different kinds of stem cells. And then they are also resistant to therapy. So these stem cells are very aggressive. Okay, they make some cells which are extremely aggressive. They will form metastasis. Here they have changed even their phenotype. Remember, androgen receptors have disappeared, something else has happened. So they have changed their phenotype. Now you give this kind of stem cell, see, they are all originating from the same parental cell. How many different kinds of things they have done to themselves. In this case, this is not even forming tumor, and this is forming tumor, but look at that, so many different kind of heterogeneous tumor. Right? Here you have given chemotherapy and radiation therapy, most of the cells died, only 1 survived. But then this is again going to make different kinds of cells. So when you have different population, if you kill one, the others survive. If you kill the others survive. If you kill the all others survive. Right? So that gives them infinite quality. So these are the properties of these cancer stem cells. Cancer stem cells the hardest to treat, okay, because they can acquire different things. They can renew themselves. They are resistant to almost all sorts of therapies, okay. Now immune evasion can also occur due to epigenetic modifications. So let me give you an example, or let me ask you an example. Give me one example by which epigenetics can lead to immune evasion. So let me remind you, let me give you a clue, and then you connect the dots, right? So yesterday I told you about PD1 and PDL1. Okay, so let me remind you about PD1 and PDL1. So T cells are the fighter cells. T cells are the immune cells, right? So their job is that anytime there is infection, they will go them, they will fight it off, right? So they get so charged up when there is infection that they are supercharged. So initially it is great to be supercharged because they go to the side, they defend us and everything, but they are still too much momentum going on. So if they're on for very long, if you're working in very high speed, it is right, so it will lead to autoimmune disease because they will start attacking that because they are in that fighter mode. So that's when APC cells come and they quieten the T cells. So they have these receptors called PD1 receptors. So if you don't touch the PD1 receptors, they are in the hyper mode if I am a T cell. Okay, so these AP1 cells will come and they have a ligand for PD1 which is called PDL1. So if they come and bind there, I will immediately become quiet. So tumor cells, what they do, they are not supposed to have PDL1, but they will express PDL1. So anytime T cells go close to them to kill them, they lock horns with them. And T cell thinks, oh, I'm supposed to be quiet, and they become quiet, and there is no tumor killing. So now apply an epigenetic mechanism. How can you use this using epigenetic mechanism? Anyone?
Tumor cell can, like, in the tumor cell, so there will be acetylation in the PDL1 gene.
Exactly, and now it will be overexpressed, right? Great. Give me one more mechanism, again for PDL1. What else can happen? Hypomethylation of the promoter region of PDL1, right? So you guys are all with me. So you can think of many epigenetic mechanisms just based on whatever we have studied so far. So it is not surprising that immune evasion can occur due to epigenetics. What is epigenetics? Oh, you have perfect DNA, but I have the control. I will decide which protein is going to be expressed and which is not going to be expressed, right? So DNA is there, but I am the decider of the DNA, right? So that is what it is shown here, that there are many different mechanisms, okay? So that is what is craved from T cell thing, right? PDL1, PD1, okay? That can also lead to resistance to immunotherapy through PD1, and I told you briefly about CTLA4 yesterday, okay? So this one is, so you know what happens, our immune system is surveying our whole body at all times. And anytime it says anything which is out of place, it will recognize it and it will eliminate it, and we are safe. When the enemy is very strong, it will fight, it will fight. Sometimes we'll get fever also, we'll feel down for days. That is immune system fighting off the infection, right? When you get cold or something, you are so drained, you don't have energy, your immune system is fighting the virus of cold, right? So that is why it takes too much strength, right? And our body is inflamed and all these things. Everything comes with a cost. When a lot of fighting is going on in the body, our body is drained. When you get a vaccine, we all get fever for a day or two, right? Most of the people prefer to have vaccine on a Friday so that if the fever comes, they have the weekend to sleep it off, right? So, and we have to plan our vaccine too, that we assume that we'll get fever. So we decide accordingly towards the end of the week so that we can at least get rest in case that happens. Okay, so but then the cancer cells are pretty smart. So even after the fight, they quieten the T cells and the tumor grows. Okay, and then it metastasizes. And then you can also plan the drugs. But then even with PDL1 drug, right, it is such a good drug. But eventually cancer cells will develop resistance towards immunotherapy also, okay? If you are attacking PDL1, PD1, they will express CTL4, something else, to evade that thing, okay? So that was immune evasion, okay? Now this is showing you some more immune. Do you know what is innate immune evasion versus, can anyone tell me that? Innate and the other one. Tell me what is innate? Intrinsic immunity, very good. So suppose we are attacked with something, our body, heaven forbid, if we hurt ourselves, right? Immediately macrophages come in and they do a lot of things. Blood clotting happens and macrophages come up, they eat all the garbage. That's innate immunity. And what is the other one?
Acquired.
Acquired immunity. And what is the meaning of acquired immunity?
We acquire from the external factors that are.
And then what happens?
We generate memory cells.
Yes, that is the main thing. Acquired immunity is not immediate, so that cannot come into play. Depending on what we are infected with, then our immune system will digest that foreign material, then the antibodies will be made against those foreign thing, then the whole thing will be cleaned up. And then the beauty is that they will save those B cells, the antibody producing cells, for future also. So we will always have the memory in this lifetime against that disease. So that is why when we were vaccinated, when we were 10 days old, we are still resistant to that because we still have the memory B cells there. So we will never get that disease unless the virus changes its thing. Okay, so that is what it is shown. Okay, here, so T cell trafficking. So you can actually, so this is evasion. Cancer cells antigen release, cancer cell antigen presentation, priming and activation. These are the things, okay, these are the things. So you can actually develop therapy against cancer cells. So cancer cells sometimes release specific proteins which are specific to cancer cells. If you can recognize that, you can develop a vaccine against it, or you can develop T cells against that antigens also. But then what will happen when you give the therapy, cancer cells will stop expressing those proteins. So now they will evade that also. So it's a constant fight. Okay. So then look at these epithelial cells. So they are changing their phenotype. Now they are becoming mesenchymal cells. This is EMT, epithelial to mesenchymal transition. So if you give drugs for a very long time, now they have changed their thing. They are very motile. They can squeeze through blood vessel. They can squeeze through small holes. They will go somewhere else. They say, okay, this room is very hostile now. Let me change my thing. Let me sneak through the door. Let me sneak through this window, become very thin, very flexible, go out and make my colony somewhere else. And then move from there and make somewhere else. That is metastasis. Okay, they find a new environment and they just colonize there. Okay, and so these are different proteins which are involved in that. Don't worry about this thing. But the more mesenchymal they are, the more aggressive they are, and the more motile they are. And more motile, more invasion, more metastasis. Okay, so it is showing that you have EMT signals, and see some of the cells, they escape, they come in the blood vessels, they come out of the blood vessel and they form a tumor at some other place. So when someone has liver cancer, they metastasize in the lungs first. When someone has breast cancer, those metastasize in the bones. Okay, so different kinds of cancer, they metastasize at different places. Okay, one time a student of mine, she showed me, so I'm not a doctor, right? So I'm not in touch with cancer patients, which is good for me because it's very painful to hear that, right? But in that case, so she had a relative who had cancer and it has metastasized. So I know all the principles. So she was from a different country, so she asked that person to send the PET imaging scan, and I saw that scan for the first time with my eyes, the actual data, right? Not from a textbook, but actual data of a person. And the cancer had metastasized in all over the body, like here and here and here and here and here. And that was very painful to see. You really pray that you don't have to ever see that thing, I mean, unless you are a doctor. But it was like, it can spread all over the body. And sometimes what happens is that we have so many drugs that we prolong the life, but it is not worth prolonging when it has spread so much. You know, I mean, we can say that, but for family members, every day counts. You know, you really want to prolong the life of your loved ones. Okay? And so this you already know. You have already given me ways by which it can lead to therapy resistance. Okay, so that was the therapy resistance that can actually occur in many different ways. All right. Now I answer your question. How do you know, right? So chip sequencing. So there are DNA methylation arrays and bisulfite sequencing, which will tell you DNA methylation exactly. ChIP-6 you have probably heard. It will tell you about histone modification. ATAC sequencing will tell you about chromosome accessibility. Please send those things to the students today, and I will send you today, provided I have access to this thing. And you can even do single cell epigenomics. This is the last slide, I think. Okay, functional, this thing, we'll talk about this. Okay, just I think 2 more minutes. So DNA methylase inhibitors are already in clinical trial, and some of them are approved. They are histone deacetylase, methylatase, and there are combination therapy of epigenetic drugs with immunotherapy or targeted therapy, and they are BET inhibitors. So what are BET? This is very important. So bromodomains are the ones which bind acetylated histone. So if you give BET inhibitors, these acetyl groups are exposed and they can be removed also. Okay, so BET inhibitors. So BET proteins are epigenetic regulators that bind to acetylated lysines. Okay, so if you remove the BET group, sometimes the acetyl group is too exposed and they can be deacetylated. And if they are deacetylated, the chromatin will close. Okay, so that way you can prevent activation of oncogenes. Okay, I will stop now. Just so this is, you guys are going to get this presentation. So if you have any questions, I can answer it later. Okay, all right, guys. So it is tea time. All right, let's have some tea, and then you guys can ask me more questions. Okay.